Hello, good afternoon, sir.
Good afternoon, sir. Good afternoon. Shall we start our session,
This is Varma from Srijitra.
sir?
This is Varma from Srijitra.
Yes, sir. Welcome, sir.
Good afternoon.
Thank you for your presence.
Welcome back to ICMPMA 2022. We now move on to the fifth session of the conference. The topic for today's session is biomaterials. To chair this session, we invite Dr. Jayachandran K., the professor in the School of Biosciences, Mahatma Gandhi University, Kottayam. He obtained his PhD in biotechnology from Cochin University of Science and Technology. He is a former member of the State Environmental Impact Assessment Authority, SCIAA, Kerala, former member of Syndicate MG University, Kottayam, former Senate member of MG University, former director, College Development Council, MG University. He has been acting as the honorary director of Inter-University Center for the Studies in Science of Music, IUCSSM, since November 2015. He has over 22 years of research experience and has over 63 publications in reputed SCI journals and has delivered 36 invited lectures in several national and international conferences and authored 5 book chapters. At present, 12 doctoral students were awarded PhD and 8 research students are working under him for doctorate degree. His research interests include bioprocess technology. Dr. Jayachandran K., we are honored to have you, a personality as esteemed and experienced as you, here to chair this event. Dear sir, I request you to start the conference.
Thank you for the nice introduction. Welcome to all. I sincerely welcome our guest, respected Professor Harikrishna Verma sir. We are in the fifth session of the International Conference on Materials, Properties, Measurements and Applications, organized by Fatima Mata College, Kollam. This section is in fact dedicated to biomaterials, and we know how significant the biomaterials are. Particularly in the context of toxicity compatibility, particularly in the context of the emergence of nanotechnology. So we have 3 invited talks scheduled in this fifth session. In addition to that, we'll also have a plenary talk. So we begin with invited talk by Professor Harikrishna Verma sir from Srijitra Institute of Medical Science and Technology. Once again, I sincerely welcome you, sir.
Thank you.
Introduce Dr. Verma. Dr. Verma is presently acting as the head of biomedical technology wing of Sri Chitra Institute of Medical Science and Technology, Trivandrum. He did his PhD from Regional Research Laboratory. Now we call that as NIST, National Institute of Interdisciplinary Science and Technology. He got his PhD in 1993, and then he joined at the Institute of Sri Chitra Institute of Medical Science and Technology. He is engaged in the research and development of various bioactive ceramics and technologies of bioceramic based bone implants developed in the lab, and that has been referred to many industries. The product is currently available in the market under the trade names B. Austin, and there are similar such items available. So he has been instrumental in translating his research for the benefit of the society. He did his postdoctoral work in National Industrial Research Institute, Japan, during 97-98 as an STA fellow. He was acting as a visiting fellow at Queen Mary and Westfield College, University of London during 94. He is a recipient of Indodad Fellowship. He has visited Germany in the Technical University. He was a senior JSPS fellow at AIST, Japan during 2002. Currently, 2 international collaborative programs are running with University of Dresden, German, and Osaka City of University of Japan. He has published more than 170 research papers in international journals. He is having 15 Indian and 1 Japanese patent, with more than 4,000 citations and with an H index of 37. He is also the recipient of many national and international awards. So we have a very good, very eminent personality with us, a good scientist, a scientist who has translated his research for the benefit of the society. So with his introduction, I request Dr. Verma to deliver his talk on search for ideal biomaterials. Dr. Verma, please.
Thank you. Can I share my screen?
Yes, yes, it's there now. Yeah, it's visible. Please.
Has come? Sir, is it visible?
Yes, sir, it is visible. I think you can move.
Hello? Okay, I can start. Just 1 minute, sir. I just switch off my video, and so that bandwidth will be better. It is not going into the. Okay. Can you see that? Can you see? Hello? Hello?
I
Is
think
it audible, sir?
it's audible perfectly, sir, but the slides are not coming. I think it's not.
Is it coming now?
No, not to me now. Even now, it is not coming to me.
I'm just sharing.
Yes, sir. Yes, sir.
Is it coming? Is it coming?
No, no, no. Maybe. No, no. It's not coming, sir. It is not coming.
So what I just do once again? I just once again screen share. It's always a problem. I'm sorry.
It's okay, sir.
Is it coming, sir?
No, no, sir. No. Sorry, no.
I'm sharing. It is showing also.
Let us inquire with other participants.
No,
We are
sir,
not
we
getting
can't
it. I'm
see.
sure, isn't it?
No, sir,
We are
we
not
can't
getting the
see.
slides.
Yeah.
Organizers, please
Yeah,
look.
sir, we can't see the slides. We can't.
Yes, yes, sir. Please help, sir. Please help Verma,
I'm,
sir.
I'm just I'm sharing.
Yes, sir.
Why that? I just once again doing. Sorry, this is always
No,
a
no, sir. No, no.
maybe due to the bandwidth problem from my side, or I don't know.
Yes,
Hello,
sir,
sir.
we can.
Yes.
We can see now.
Yes, sir. Perfect. It is coming up, sir.
Yeah.
Thank you, sir. Please, sir. Please.
Hello, can you hear?
Yes, sir. Please, sir.
Yes.
Yes. Slides are visible. Yeah, you are audible perfectly, sir. Please.
Okay, so I'm from Sri Chitrathama Institute of Medical Science and Technology, and I'm heading the biomedical technology wing, which we developed several products, which we have some of the flagship programs or products like valve, heart valve, mechanical heart valve, blood bags, and several other products which is used by and commercially available. So the thing is that when you really look at the, for example, a development product, one has to go through these type of steps, like, first product design and a number of evaluation, not only the physical as well as the chemical as well as the biological evaluation. Then we go for a systematic clinical trials and before even really go to the market. So it's a very, very involved steps and so I start with this one. We have been using several materials. This is I have taken from a popular book on ABC, so handbook by Reader's Digest. You can see that there are several materials like metal, ceramic, and polymers have been used for this type of reconstructing a hip joint or a knee joint. And we choose materials of appropriate thing which can be tolerated, as we all know about the toxicity and the biocompatibility, and one has to stay there for a long years in our body, and still we have a lot of problems. It is not ideal as compared to our original implants. So so the thing is that always we search for better materials. So the thing is that I always think about this famous person who is from Iran. He in the Athens Olympics, and he has lifted more than 350 kg. So many times, how these strong people, with a very, very soft materials, which is composed of a human body. And you can see that in our legends also, we know that the Bhima hit the Duryodhana in his this gluteus maximus, which is a very strong muscle, which is able to hold our whole body, as we have seen the last slide. So the thing is that we know that these type of very, very strong biomaterials, which is helping our body. And when you look at this in microscopically, you can see they are very, very strong with respect to their bundles of collagen fibers arranged in certain fashion, and which has got and we can have the simile of this type of ropes, filament and filament, and in the ropes like that. But the thing is that compared to these synthetic materials, we have some of the properties like the regeneration, which especially in the muscle, you can see that these type of specific cells like which is myocytes, which is there are so many small cells called in the quiescence. They are just keep it there without any keep it quiet. But whenever an injury is happening, this will be recruited and repair the whole system. So this is one of the most challenging thing. As a biomaterial scientist, I always like the nature has got these type of exotic properties. So as a person working in the last many years in the area of reconstruction of bone and teeth, all this one, we encounter a number of these applications which requires either synthetic materials or our own tissues. So if you look at the evolution, the first generation, second generation, third generation, and I can show some of this application. So we know this first generation from the very 7th century Mayan civilization, they have used ivory. So this is the type of knowledge at that time very strong, which is an inert material. It just serves the purpose. But when then later, you see in the in last century, we have seen the great person like brain mark implants. This was a revolutionized the implantology in a great direction when he has used this titanium implants and its beautiful property of osteointegration, which the natural bone is attaching to a synthetic material, which gives the strength. And nowadays the whole dental implantology is based on these implants, which has got an excellent property of osteointegration, which it attaches to the natural tissue. And there are several mechanisms where which in a host, an implant can be bound to a tissue like a bone through different calcium oxygen bonds or calcium selenol bonds, like this famous Larry Henge's glass, which is a bioactive glass where SiOH helps in this type of bending without having any fibrous tissue encapsulation. So this is attached. So this is one of the most important property of a successful material which can be integrated into the natural tissue. So then when we saw the second generation implants, not only they serve the purpose of the implants like, the drug-eluting stents where which, so we can have this type of antiplatelet type drug-related platelet, which is eluted from the surface of this one through the polymer coating, which gives not only the purpose of the increase in the diameter of the blood vessel, but also the prevent the restenosis, which make this plaque formation again and again. And then we have seen the third generation type of tissue engineering where which, whether we can have these type of synthetic tissues implanted and then we can regenerate the whole thing tissue back with the help of growth factors as well as cells, your own cells or the cultured cells over these tissues. So we are almost trying to mimic the third generation of implant. So but the thing is that, when some of the very, very severe problem, because it is a repair, remodeling and resorption is very challenging. And we know the story of magnesium as an implant material which completely failed because of it. The resorption generated so much of unwanted products. And so it's a very, very challenging. So my talk will be, I will be restricted on some of the graft substitutes and how these things have been really coming to the clinical and the new applications, and still we search for the ideal material. That is my whole message, because there is no ideal material. So in an Indian scenario, we have been lost huge number of orthopedic and especially for the dental applications, we depends on many of the implants and huge numbers. So there is always a demand for this material in India. And you can see that a natural bone healing process will happen itself if you properly stabilize us like this. It will blood formation and then the bone will attach back to the together. But when we have larger fractures as well as tumors and as well as cancer and many other deformities, we may require a graft material. So people initially, in the case of bone, people used to use your own tissue, like an own bone, then allograft from other persons or other animals, xenografts. So many things are tried, but this associated immunogenicity problem, infection and rejection. So ideal may be your own tissue, but the problem of you always create a donor side morbidity where you harvest from each side, which creates a lot of deformity in that place and the pain in that side. So if can we have the ideal material? So this is the one of the things on bone. The donor side morbidity is very, very large, and for the larger tumors, larger resection, still we cannot take enough tissue from your children and elderly and low volume and poor quality. So it's quite difficult. So that is why we go for synthetic materials, and people tried all this on matrix plus mineral alone, mix matrix alone. But obviously, an ideal graft should have all the properties of a your own bone, that is, you are called a autologous bone. And so in my laboratory, we have as a bone is a calcium phosphate mineral containing tissue. We tried all sort of compositions of calcium phosphate like hydroxyapatite, tricalcium phosphate hydroxyapatite mixture, or bioglasses, or even cements. Calcium phosphate mixed in certain ratio, which can be using as a cement. It's a putty form which can be used for in the powders, blocks, dense blocks, or coatings, or metallic. And so many type of materials have been tried and successfully. And these are, we have seen this one, why we go for this. Any of these materials should have a property of attaching and to the natural tissue. So, and these are some of the interesting things. You can see this white area is a synthetic material, and the yellow one is the new bone, which slowly integrate and slowly eaten away the synthetic material and completely engulfed with the natural tissue. So these are the type of histopomorphological pictures which I am showing of some of these material which really heavily remodels and back into the natural tissue. So we know that hydroxyapatite is an ideal material. It's a very similar to our bond structures, and there are other different type of this one. I will just show these are some of the real implants which has been used in the human, and as well as in the some of the companies which they make, for example, in the form of granules, in the form of wedges, or in the form of cylinders. And then you can see some of the real clinical application of this one. When we have large bond loss, you can use this type of bond chips or the powders which is being already in the clinical use. Or even you can see that these type of buttons, which can be used for closing these type of surgical holes when you undergo neurosurgery procedures and other things. So you can see this one. So one has to really appreciate this one, how the surgeon will be happy to see this one. When after the surgery you cannot even distinguish whether this patient, the second patient has undergone a surgery also. And we can see that this type of digital subtracting angiogram, this is exactly like your bone. You can see the blood vessels without having intervention. If you use a metallic thing, it will be interrupted, and you cannot get a clear image of these type of blood vessels in a angiogram. So we did many other these type of implant. For example, this is a very specific clinical case where when it's a vertebral column has been expanded, and you can see that this almost more than 2 centimeters, where the spinal cord is going through this hole, which where the patient was almost paraplegic, or she could not walk. But after the surgery, it was relaxed and she could walk from the hospital. So Dr. Menon, my collaborator from Amrita, has done it long before. And these are some of the structures. Really, this is the type of reconstruction in the maxillofacial area, where you can see a large bone which can totally change the shape, as well as aesthetically one can really do these type of reconstructive surgeries with purely synthetic hydroxyapatite implant. And then we change from the ratio of calcium phosphate change, so that we want faster resorption. So rather than hydroxyapatite stay there for years, maybe 3 or 4 years only, the resorption will not be, but it serves the purpose. But then we change it into compositions of different type of fast resorbing material. You can see that these are a calcium phosphosilicate material, which you can see that it has been by 6 months it can be completely resorbed into the natural bone. Whereas here also you can see this was a large gap, but after filling this gap, and one can really see that it goes. And even larger cystic cavities, where ilia crust, where with large bone, you can see that this is a huge bone loss here. You can see that how they can use these type of blocks, and by 1 year you can see the completely regenerated ileum or ilia crust. You can see that this is a huge gap was there, and it is completely reconstructed. So, and this is again the X-ray picture of such implants, where it's purely synthetic materials. We can accelerate the growth, as well as remodel the fastness of the remodeling. You can see that here also the block was here, and after 1 year you can see that it completely regenerated the bone tissue. And this is the comparison where I'm saying if you use a pure hydroxyapatite or a non-resolved, it will be there for years. But you can see that the radio opacity, where here you cannot even distinguish whether it is filled by a synthetic material which is absorbed into the natural tissue. And this has been again used for different applications, like the maxillofacial, where with a large resection where it is happening, and you can use this one for the total reconstruction of the mandible or maxilla. And these are some of the products which we could put in the market, and then we search for new areas where we could use this synthetic cements like calcium phosphate cement in different calcium phosphate ratio. This is called HAP cements, which is almost mimicking like a natural bone mineral. You can see the bone mineral as well as the injectable cement which we prepared. It's a wise crystallography which is almost similar, and it can be used for many applications like these type of cavities in the dental as well as the orthopedic area. So the thing is that these are again the picture, you can see that these are the completely cavity was there, and after 9 months you can see this is completely filled by the natural tissue. Here also you can see the natural tissue. So then we are, as I told you, the second generation implants, it is not only serve the purpose of a filling, but we can use this similar type of drug eluting one, where which we are using some of these beads having nano size porosity, which can hold the drugs or like antibiotics, which is used for this treating this type of infections, where which you not only the implant will be there, but it elutes some of the drugs into the site, and you can see that whole area can be filled faster. And you can see these are the real filling of these granules, and this whole cavity has been slowly completely healed and with a natural bone tissue. Whereas, so that I am saying is that not only the implant, but also we can use these drugs or growth factors along with your thing with which can really supply into the required area of application. And this is one of the major challenges which we have, a large bone defect, whether we can really regenerate some of the large resection due to the tumor, bone tumors and other things. You have 10 centimeter long bones like femur or a tibia. So we are working and with good success this type of cylindrical thing where which we have a growth factor and own cells, we can, this is called the tissue engineered cells, which is really happening, and this type of large porosity where which it can hold, and you can see this as an implant in a God model, and you can see that this is the actual tissue engineered graft, and you can see after 6 months, the whole area has been completely bone regenerated with the natural tissue. So you can have the tissues like natural materials like hydroxyapatite or bioglass along with the cells and growth factors. One can really use a synthetic counterpart of the natural tissue, which can be really good, see the very good results when the remodeling these type of bone implants. And this is again the X-ray show. This was the actual implant, and after 6 months, you can see that whole tissue has been completely regenerated. And this is the type of electron microscope which we have a ESM, environmental scanning electron microscope, where we can see live cells. These are the fibrous osteoblasts which is growing into the porosities, which is almost mimicking like a natural bone. And this is the actual the human mesenchymal stem cells cultured over this graft in clinical center at CMC Vellore. And Dr. Burja Mathri is a fantastic orthopedic surgeon. She has been doing this work. So, and I won't take more time because it is 2:30. I don't want to this one. So these are the type of things which we are really look for. We have these type of femoral heads or a total vertebra. These are all the things where which we don't have anything which is best material. But once you have broken, you go for a synthetic or a metallic process. But can we design these type of totally synthetic, which is much more compatible with the tissue? So these are the other challenges, like whether we can have this type of clefts, whether bone can be. Now only we are stitching back the soft tissue, but can we have the maxillary bone regenerated back? Or these are the large bone defect, whether we can really use these gaps using this type of tissue engineered system, where which we can fill the gap with the natural tissue. So there are other applications of these type of porous materials which can be used not only for the implantation but the cancer research, where which one can use these nanoparticles, where with magnetic nanoparticles containing which can be released. The drugs can be released by an application of an applied field, and there are other coatings which is also again for the dental implant, which has a specific property of osteointegration as well as drug eluting property. So this is a nanoporous tubes of titania generated or dental implant, which can carry drug and other things. So we will look at for the these type of drug eluting implants, metallic implants also, and these are some of the results of this one. But I stop here, and this is again the same type of stair, where which by putting more of these type of growth factors, one can really see that the bone. This red one is the new bone compared to the bare implant. This is the second is the bare implant, whereas the red one bone can more bone growth happening in such thing. So I stop here. There is no universal panacea for this type of thing. We require a number of new applications and the effective interaction with the clinicians and the scientists. That is the ultimate success of such this one, which can really go to the translational stage. And I stop here, and thank you for your patience here. Thank you.
Thank you, sir. Thank you very much. It was a very nice presentation, very good information. Really happy to note all those translated products. I think we can have a couple of questions now.
Yeah, sure. Sure.
Yeah. So this is a golden opportunity. We have Dr. Verma with us. So you can have 1 or 2 questions. Dear audience, please.
Hello.
Yeah. I think nobody is coming up. So if you have any question or if you have any clarification or if you want to consult Dr. Verma, please do that now.
Sure. I'll be there. I'll be always there to help.
Yes.
Thank you very much.
So I think as we don't have much questions now, we will thank you, sir. Thank you very much for your for spending your valuable time for this international conference. We are really fortunate to be here with your presentation. Thank you very much, sir. So with your permission, we are getting into the second talk, invited talk of the fifth session. So we have, I think, organizers, shall we move to the second invited talk? I'm sure Professor Virginia, Dr. Virginia is there with us?
Yes, sir. Yes, sir.
Yes, okay. Welcome, Dr. Virginia. Welcome to this fifth session of this international conference.
Hello. Yes, can you hear me?
Yes. Yeah.
Yes.
Good afternoon. Good afternoon. Thank you for your presence. So we have the second talk of the session 5, material toxicity and manufacturing challenges in microfluidics. So we have Dr. Virginia with us. Yes, good afternoon. Nice to see you in online mode. So we have Dr. Virginia. She's an associate professor in bioelectronics at the University of Leeds. She graduated, she could get her graduation in 2004 in electronic engineering. Yeah, it's very nice. From the University of Pisa. And she got her PhD in PhD from University of Genova and Italian Institute of Technology in Italy. That's great. So she was an exchange researcher at the Department of Life Science, Medicine, Bioscience, in Japan, and thereafter she worked as a postdoctoral research associate in USA from 2011 to 2015. In 2015, she became the research assistant professor, and then she moved. I think she has also participated in and completed many medical startup programs, and she has served as CEO for 2 years. Her research was sponsored by European Research Council and Wellcome Trust. So that's great, no? Project being sponsored by Wellcome Trust. So she's also interested in the pathophysiology of infertility and pregnancy failure. Oh, that's great. And in the development of micro and nano tools to evaluate the effect of toxic and exposure. So we have a very good personality with us. Thank you for your kind presence. So now I request Professor Virginia to deliver her lecture on material toxicity and manufacturing challenges in microfluidics. Thank you, please.
Thank you very much for the kind introduction. So I'm going to share my screen. Please confirm that you can see it. Can you see my screen?
Yes, very good.
Perfect. So thanks again for inviting me. Thank you for, I want to thank the chair of the conference, and it's great to see this very big audience for this hybrid conference. And so it's a pleasure for me to be connected from UK and be able to listen to these exciting talks. So I just want to tell you where I'm connecting from. So I'm calling from Leeds, which is in the north of United Kingdom. So it's quite different setting. It's a nice city. So please, as soon as possible, once traveling would be easier, please come and visit. I would be happy to invite and show more about my research here. At the moment, I'm sitting in the School of Electronic and Electrical Engineering, sort of down here. So I'm glad the connection is nice and you can hear me well. So my research, as from the introduction, is about microfluidics. It's quite basic, but it's always good to set the right definition of microfluidics for the ones that are not familiar with that. So microfluidics is related to fabrication and use of techniques that are in the scale of hundreds to a few micrometers. So we want to create in vitro system where we can host cells or any organism or molecules that are below the size of 100 microns. In this case, if we think about the volumes that we're handling, we're going below microliters, because microliters are more or less, you may have become familiar with lateral flow testing for COVID recently. And in that case, the drops that we are handling are in the order of tens or hundreds of microliters. Based on the size and the scale, in this case, we are handling things that are much lower volume, as here in this cartoon, droplets of aerosol from COVID. So it's quite difficult and complicated to manufacture the system, but nowadays we have several techniques to do that. And I'm very concerned, I'm very interested in looking at and monitoring the biocompatibility of materials that we're using and any changes that we are doing during the manufacturing process and how they can affect the viability of our natural components. So microfluidic is not new or artificial. So the first example of microfluidic that we can find in nature is the vasculature, blood vasculature. Because the typical size of these of capillaries, for example, goes below 10 microns in section. So this is an inspiration for us, thinking when we study and when we learn how the fluids are moving into a microfluidic compartment. So nowadays we talk about microfluidics in terms of studying and understanding the fluidic, but also, as I said, manufacturing system. And we are used to adopt for science alternative in vitro system, like the one that you see on the left, which is a standard commercially available microfluidic device in polystyrene with a straight channel, with a typical width about 0.5 millimeter. In this case, the amount of fluid that we have inside is below 100 nanoliters. So we are already going down very consistently. On the right, instead, you see a bit more complex structure that effectively recall the microvasculature, the natural microvasculature. And again, we're now able to recreate this sort of systems. So this is quite exciting to recreate in vitro models for specific disease like neuropathologies, or in my case, I'm very interested to recreate specific organs. And this is because microfluidic has been proposed in the last more than 10 years for an alternative method in vitro to test drugs. We know that drug testing is a quite long process. So if we look at something from the past, from 1928, when penicillin was first discovered, it took then more than 20 years to effectively be tested and used on large scale and manufactured safely at large scale. This was many years ago, but recently we have seen instead a rapid development and design test of a vaccine. And the rapid testing, of course, was the speed was related to the availability of lots of funding and a global effort to find new therapy, but also because of existing data already on the method and approach to design the vaccine. But still, testing is long and can have some unexpected result. So in general, in the last 5 years, it has become obvious that pharmaceutical company risk to waste a lot of money because of low efficacy of drugs once they arrive into the market. So if we look quickly at the drug testing process, in general, testing and design of a new molecule for therapy start from several thousand of compounds that are then, first of all, assessed for safety and for efficacy in vitro using cell culture, primary or cell lines that are quite stable and quite controllable. Once the compounds that are successful at that stage are then tested with animal models. And only at the time when the preclinical trials are completed, a few compounds or a handful of compounds are usually assessed for safety, first of all, in clinical trial with humans. This full process, so from the drug development, so from the drug design to the effective validation in humans, can take more than 10 years. And more importantly, it can take several billions, so a lot of money for pharmaceutical company. And as I said, unfortunately, we have seen many compounds producing unexpected side effects or not performing as expected. So the idea, the first use of microfluidics and organs on chip has been proposed to improve the efficacy of this drug testing process. And this is to summarize. So the idea to use, the standard use of in vitro 2D culture is quite useful because there is a high controllability and reproducibility of the system. It's a quite high throughput approach, but is not physiologically relevant because we have cells that are completely outside their natural environment. The microenvironment with all the 3D structure is not present. Using animal models instead, we have the opposite. We have a very high physiological relevance and the complexity of the full human body with the communication between organs is recreated. So we can assess side effect, we can test the targeting efficacy of the drug, but it's very difficult to control an animal model. And it takes a lot of effort, time, and cost to make it to have reproducible results. So 2 approaches have been proposed are currently used. One is to include 3D cell culture, so possible matrices and scaffold, for example, that can recreate in vitro the real mechanical and chemical cues that cells are exposed to in vivo. But again, these are not, the majority of the case, they are not, they do not fully recreate the complexity of the organ, the functions, and the mechanism underlying the characteristic of an organ. So organs on chip in 2012, there was a huge, one of the first huge program funded by the National Institute of Health in US, aiming to recreate a full human on chip. So several organs automatically perfused and communicating, so that the effect of a drug, the release of a drug of an organ, could have been tested, and the effects all around the body can be recorded with a huge control and a very huge improvement in terms of understanding pharmacokinetic and pharmacodynamic. So the advantage of these organs on chip is that we can effectively use, in most of the case, patient-derived cells. We can replace or reduce the use of animals, so which is quite important for ethical concern, but also, as we discussed, for the limits of these animal models. It is quite high throughput because, as I will briefly describe, the method to manufacture this system is consistent with high-scale production, and the volumes that we are handling are, as we discussed in the first slide, very low. So this is overall quite cheap, meaning not expensive method, which is based on use of plastics. So this allow me to go to the main point of my topic. This is quickly an example of one of the best example of organs on chip, which is the lung system that was developed by Professor Donald Ingberg at Harvard University. In this case, you can look at signaling between two different cell types and recreate the epithelium in the lung and in different upper and lower respiratory system, and look at the communication between epithelial cells and capillary cells, so endothelial cells, in a lung model. The characteristic of this system is that it's possible to stretch this culture in a very highly controlled manner, so that the cells are effectively showing the characteristic phenotypes, and it's possible to recreate the main function of this epithelium with a very tight barrier, and then test drugs for crossing of molecules and bacteria. So this video, I hope it will work, show you an epithelium with some cilia, and this is a culture that has been grown after about a week. So thanks to the mechanical stimulation and the eye control of the co-culture, it's possible to effectively see the specific morphology and have not only the formation of these cilia, but also effectively see their function as moving and helping mucus to flow through the system. So my interest is related to organs on chip to recreate female reproductive system. So I'm really interested to have a new model and a more efficient model, effective model to look at fertility and to look at fertility treatment in a certain way. So here you see the female, uterus, and the different organs and tissues that are part of that. So in my lab, we develop organs on chip that recreate parts of this, so that we can look at cyclic changes in the morphologies of this tissue. We can look at changing in function of endometrial cells, for example, during a menstrual cycle. And we are working, as I will show you briefly, on a system that can support growth and development of eggs and embryos. The key point of this, as for the overall for organs on chip, in the organs on chip community, is the possibility to use cells from patients. So in my case specifically, I can access and I can isolate the different cell types from placentas, from C-section, so plant C-section, and blood samples. So we have an ethical approval to collect these tissues when women delivered at the St. James Hospital here in Leeds. And we can extract, we can isolate quite pure culture of endothelial cells, monocytes, stroma cells that are basically the fibroblast forming the endometrium, and epithelial cells. And we can then use them as main blocks to recreate the endometrium, fetal membranes model, and other example. So these are the three main platforms that we use for toxicity tests and for looking at specific disease affecting the reproductive system as endometriosis or infertility. So I don't have time to go through the different platform. I will only talk about the embryo on chip, but I would be happy to talk about it or separately if you want to know about it. So the embryo on chip, here a very brief description of how an embryo develops in vivo. So in the natural system, what happens is that every month an egg is released by the ovary, and if the fertilization occur, then the embryo start to change, showing duplication from 1 cell to 2 cells, then 4 cells, 8 cells, and moving to what we call a blastocyst. So this process takes from 3-4 days in animals, in mice, take a bit longer in humans. If successful, if this embryo develop at the right time, as shows the right morphology, the right characteristics, and if the endometrium, so if the mother if the endometrium has the right is in the right condition, then an implantation will occur and a pregnancy will start. So I've been thinking, and we've been working, designing a system that can mimic this tiny tube, the fallopian tube, and support this development. This is important because a treatment that is available nowadays and is quite used by millions of couples that are unfortunately not able to conceive naturally is in vitro fertilization. And in this case, eggs are collected, so are retrieved from the patient and combined with sperm in vitro, grow for, as we said, about a week, and then transferred back into the uterus to start the pregnancy. So if we want to do this, we need to have the appropriate substrate. So currently we use Petri dishes, but we are working towards the replacement of these Petri dishes with a microfluidic system. So here you see an embryo growing in vitro, and you will be able to see in this short video the changes going from 1 cell to up to 8 cells, and then the compaction that makes sort of morula, a sort of blackberry shape. Then once the blastocyst form, at the end, this blastocyst will expand so much that the external shell will break. We say the blastocyst hatch, and the embryo effectively try to attach an anchor at the bottom of the dish. So here you see quickly these changes, this speed up video. Now we have a blastocyst. You see the cavity generating, and then it hatch. And in vitro, it tries to find a place for anchoring and adhering. In vivo, of course, it would implant and then connect and start the formation of the amniotic sac gradually. So we want to be able to see in vitro these changes, and we want to be able to see this development up to blastocyst occurring at the right time. And this is an indication of a potential of implantation, so potential of good pregnancy and successful pregnancy. And this is also an indication of absence of toxic compound or stress due to the in vitro culture. Sorry. So traditionally, the system that is used at the moment is based on polystyrene dishes as a very common material in biology and in vitro culture, which is not just polystyrene, but is a specific IVF certified polystyrene that goes through several more specific toxicity tests. This is our system instead, that consists at the moment of a microfluidic layer of polydimethylsiloxane. PDMS is a quite common material used for fast, rapid prototyping because it has the advantages of being optically transparent. It's used and accepted as a biocompatible material. It's quite simple to use, so every student can design something like this quickly, knowing the basic of 3D design, and can then recreate quickly a microfluidic system. It has specific characteristic in terms of gas permeability, so it allows oxygen, for example, to go through the plastic and reach the microfluidic compartment. So here you see on the right a group of embryos at different stages growing inside the device. I'll show you more later on. So the very important test for us is to look at the toxicity of this material, so this PDMS device. And so we use the gold standard in embryology, which is the mouse embryo assay, where we need to show blastocyst rate above 80%, which is what we confirm comparing our microfluidic device culture in gray with the microdrop culture, so the standard control culture in Petri dishes that is shown in black. And we can assess from morphology, and we also perform PCR and other assay. We can confirm that these blastocysts are quite healthy, and from the initial group, they basically all of them, 100% of them, develop nicely. So the test for toxicity, the standard test for toxicity is positive, but we want to look more in detail into this because there are known long-term effect of in vitro fertilization. So there are some evidences not yet confirmed in the field of the effect of stress, so material possible related to material toxicity, to not precise control of pH and temperature and gas exchange in vitro that can lead to development in adults of diseases like cardiovascular disease or diabetes. So we want to look specifically at the microfabrication technologies that we use, which is photolithography, and it's a common technique based on silicon that is used, for example, for making electronic circuit components. So in this process, we use photoresist, we use solvents to dissolve this photoresist. And so we're concerned that some of these can effectively remain in the culture, or some residuals can be traced later on. The final step in this fabrication process is the addition of this PDMS, so this silicone. This step is doesn't require any solvent, which is can be quite harmful in terms of embryo and fetal development. So it should be quite safe. In order to understand what is happening, and to see if there is any, if we can go further into the detail of the toxicity, we perform untargeted global mass spectrometry, and we compare media that we kept in the empty, so media from the device that has been kept in the device for 5 days with fresh media from the bottle. And we also compare media from the full culture, so that we extract once the blastocysts are developed. And we compare these to the control, so the fresh medium. So these untargeted global analysis enable us to detect several compounds and have a putative identification of these compounds. What we can tell from this analysis is the difference in terms of different comparative different presence of compound. So we do not perform at this stage a specific analysis to quantify the amount of compound, but we just want to see if there are differences between the groups. And this is effectively what we confirm. So we can see here in this PCA plot, you see these 3 different colors represent the 3 groups. So media from the device containing embryos, media from the device that was not used for culture, and the original medium. So they are absolutely different in terms of composition and presence, different presence of compound. So we can screen more than 200 compounds that were detected in the different groups, and 700 of these are differently expressed in the different groups. So here, for example, you see that comparing 2 groups, we have upregulation or downregulation of compound in spent media from devices compared to the original medium, and spent media from culture compared to the original medium. So this is a list, and there is also a web page to go through them in case some of you is a specific expert of some of these material and want to have a discussion about it and have some understanding of the toxicity of these. I just want to mention some of these. So we effectively have been able to detect basically compounds that are a constituent of the PDMS, and are also ideally and nowadays accepted as not toxic compound, but they have not been tested with mouse embryos or human embryos. So it's good to keep an eye on this and to be able to possibly remove all of them to have a 100% clearance of this compound. These are more, and these are specifically compounds that are nowadays used, for example, for Teflon, for Gore-Tex. They are used for as diethyl phthalic acid is used for, as plasticizer. So it's commonly found in food packaging, for example. So they are accepted in general, but not specifically tested in embryo culture. So this was a first sort of investigation of metabolomics in microfluidic device for embryo culture, and we can tell that there is a very good viability of these embryos in this device, and we want to perform further testing to understand what are the possible effects of the residual. We have some methods to reduce the leaching and the absorbance, for example, of PDMS of some molecules. And we are now looking at changing the material so that we can use, for example, polystyrene. So we now produce the same device in polystyrene, and we are aiming to perform the same mass spec analysis to see if these traces are not present anymore. And once this is done, we can then hopefully translate, and it's just the last one, we can translate this into a commercial available product and move to validation and testing with human embryos and possibly move into the clinics. So sorry if I've been too long. This was the last one. I want to thank again everyone for inviting me, and happy to take any question.
Thank you. Thank you very much. Organ on a chip. Yeah, definitely innovative technology. Right. Definitely a paradigm shift. So thank you very much. Very informative to us. I'm sure there will be questions from the audience. So we can have a couple of questions now. Please, a good opportunity is there now. We have Professor Virginia with us, such experienced person, very good research work, golden opportunity for sharing.
I see.
I want the audience to come forward, please. Yes.
You got a hand from Sajid George. Yeah.
Yes.
Yeah, thank you. Excellent talk. My question is, why don't we fabricate these kind of lab on a chip using glass substrate instead of the plastic ones? Because I believe it is possible.
Yes. So first of all, it's a matter of cost. So manufacturing glass and manufacturing plastic is change definitely the cost. The other thing is the flexibility of the in terms of design. So we can definitely design quickly very small fissures in with this SU8 mold. So using this technique, while it's not that straightforward with glass. The other thing is that glass is not gas permeable, and we definitely want to have and to control the gas exchange, especially oxygen, so that we can keep the right condition during the culture in vitro.
Yeah, a related question, if I have time, is that during this kind of fabrication, I presume there are chances of formation of nanoplastics or microplastics, and now this is the key word for toxicology. So do you think an interference from these kind of particles formed during the manufacturing in that toxicity assay?
That's a great question, and it's actually something that I'm trying to look at with some colleagues at the moment. There are no evidence, there is not a lot of literature in terms of microplastics residuals from PDMS or from SU8, but it's something that I'm interested to look at because there might be. There are for sure some residuals, so micro or milli residuals due to the mechanical machining that we do with a PDMS layer. They don't usually have huge effect in vitro, but we're running now one of the many animal trials to look at pregnancy rate and fetal development, so we could confirm the biocompatibility of the full process. But that's a great point.
Excellent, thank you.
Thank you.
Yes, thank you, Professor Saji George, for that valuable question. Yeah, thank you very much, Professor Virginia. Those slides were excellent. Now, video was very informative. Thank you for sharing your valuable time with us.
Thank you.
Thank you very much. Yeah. Now we are moving to the third invited talk of the fifth session. We have Professor C. Sudarshan Kumar with us. I hope sir has already joined. Sir. Professor Sudarshan Kumar, sir, I'm sure you are here.
Yes, I'm here.
Yes, sir. Sir, good afternoon. Welcome to this fifth session. I'm really happy to welcome you. I'm really happy to introduce you. For the audience, Professor Sudarshan Kumar sir is senior professor of our own university, Mahatma Gandhi University. He was formerly the director of School of Pure and Applied Physics. I think, he took his BSc in physics from the same college. So I think we should appreciate. Now, we have a former student of the same college, Professor Sudarshan Kumar sir, for giving us an invite to talk. He took his MSc in physics from Kerala University. And after that, he did his PhD in X-ray crystallography from Indian Institute of Technology, Madras. And later on, he joined at Indian Institute of Science for his PDF. He was there in Ohio State University, USA. He was there in several other research institutes. And if I remember correctly, he has also spent, he is a co-worker of the former Nobel laureate. I'm sure I'm right, Sudarshan sir. Sir worked with Venkataraman Ramakrishna, the Nobel Prize winner who has contributed to the structure of ribosome. So with this introduction, I invite sir. Sir's optional topic area of interest includes X-ray crystallography, biosensors, structure and dynamics of modified nucleic acids, interaction of natural drugs and their functionalized nanoparticles with nano micromolecules. So with these words of introduction, I request respected Professor C. Sudarshan Kumar to deliver his lecture on, invite to talk on probing the interaction of natural drugs and their functionalized nanoparticles with macromolecules, spectroscopic, calorimetric, and computational approaches. Professor Sudarshan Kumar, sir, please.
Thank you so much, Professor Jayachandran. Of course, I am the former student of Fatima College. I did my BSc there. It was very nice. From my +2 onwards, I was there for 5 years, and I still remember a lot of things over there. You know, the beautiful days which ever I had in my student life. I remember that. And also, I thank my organizers for giving me an opportunity to present some of my work here. I will directly go to the presentation. Shall I? Yeah, you can hear me,
Yes,
right?
sir, please.
You can
Please,
hear
sir.
me, right?
Yes, sir. Yes.
Okay. I think you can see this also, right? Hello?
Yes, sir. Yes, sir. We can see.
Okay, okay. My title is Interaction of Natural Drugs and Their Functionalized Nanoparticles with Macromolecules. You know, Ayurveda, our traditional good old medicine, it uses our own medicinal plants. They have several therapeutic applications. Many phytochemical compounds which are abundant in medicinal plants like phenols, alkaloids, flavonoids, steroids, terpenoids, and so many. They are responsible for biological and pharmacological activities. So the interaction of these molecules with the nucleic acids and proteins are key process in understanding their therapeutic applications. You know, the very basis of life is based on the interaction and the signaling and the interaction between different type of molecules. That's how the life exists. So DNA is a vital molecule that is involved in several processes like transcription and translation. So one of the target for drug molecules is the DNA. So I would like to explain some of the work that we have done in that direction. So little bit of introduction, as you know, you in school classes, it's DNA, what DNA is. It has a base, sugar, and a phosphate backbone that constitute a double helical, duplex helical structure wound together in the form of a right-handed helix. Of course, you can have even left-handed DNA, which is, of course, doesn't have many functional roles. The right-handed part of DNA is the most important, which is the B form of DNA. There are two type of grooves in it. Surface, as you see, this is the major groove, and this is what you call it as the minor groove. When you wind the DNA, the duplex strand of DNA, you get two kind of hollow and a deep groove on the surface. This is where we target for the direct binding. So a little more about the direct binding interactions. You can have covalent binding or non-covalent binding. You can have electrostatic binding or groove binding, or intercalative mode of binding. Groove binding mode of consisting of two type, minor groove and major groove binding. So this is cisplatin. In this picture you have cisplatin, the drug commonly used against anti-cancer, binds on the DNA backbone. Here again you have find another kind of drugs complexes which binds on the DNA on the surfaces, on the sugar phosphate backbone surface. And then you have intercalators like donomycin, and then you have groove binders. This is what you find here. So all these type of binding, these majorly these four type of bindings are well known and studied by several people. So some of the DNA binding grooves, groove binding drugs, what we have done, you see that you have netropsin here and distamycin. You have complexation with the crystallography, and these complexed with the crystallographic DNA molecules, small structures of DNA molecules, and found the exact type of interactions. So all these, what I have shown here, 6 of them are groove binders. The importance of groove binders is that the shape complementarity. If you look at the surface of the DNA grooves, you have a shallow kind of shape which is complementary to these type of overall shape of these molecules. So the binding process is very easy for these type of molecules. We will look at what are the different type of interactions you have. The groove binders, actually you have all these type of electrostatic interactions, then hydrogen bonding interactions, water mediated interactions, and covalent interactions. All those van der Waals also possible. Stacking, sometimes even stacking you can have to keep these molecules in the grooves. So if you look at the minor groove binding molecules, drugs, that it causes less perturbation to the binding site. So the functionally cannot be, it's all right, cannot be altered. Then high sequence specificity, sometimes you find that AT rich sequence, or sometimes some other times you find GC rich sequences. So it is highly sequence specific. So we will look at this. Okay, this is what is netropsin structure, and this is what is distamycin. They are well-known groove binders, but they are anti-cancerous also. But unfortunately, they have very toxic. You cannot use it. So the first thing that we are going to discuss is the curcumin. You know that what it is. So it is antioxidant, anti-tumor, and anti-cancerous. So many properties are there. It's good old. Even it is a good old medicine for our kitchen. So we call it as grandma medicine, one of the best grandma medicines ever in our kitchen. So minor groove binders, if you look at these are the two netropsin and distamycin. You have curcumin here, and you have another molecule like piperin from pepper. So these two are also equally important for binding in the groove of DNA. Okay, let us look at it. We have some more molecules like luteolin, flavonones, then hesperidin, isofgenistein. Okay, say gingerol from ginger. All these molecules we have studied, and I would like to make some of them only. I will limit myself to curcumin and piperin, and other compounds some we functionalized with the nanoparticles. So first of all, you know, they're extremely difficult to make these molecules to co-crystallize with the DNA stretches and find out their structure becomes almost impossible. So therefore, another method that we can use is the computational way, the molecular dynamics. So the strategy that is followed in molecular dynamics is that you have different type of interactions. The exact type of interactions are known, the bond distances, bond angles, everything is standardized now over the 100 years of crystallography. So it has been standardized. So reasonably we can compute the molecular energies and their conformations. So what we need to do is from this equation, the bond distance, you need to add bond angle, you have torsion angles, you have other electrostatic and van der Waals. All these put together, you'll get an exact structure of the compound. So the strategy that is followed is initial positions and their velocities. How do you find out? This is from the initial positions are finding out from the crystallographic database. So the structures are available. You heat this to some system with some temperature and find out their velocities, and then put it back and start the dynamics process. So I don't go into those details, but the results are becoming so important. So the molecular dynamics is quite convenient way of finding out the molecular structure and interactions between macromolecules and small drugs. So if you look at, okay, so if you take the DNA curcumin complex, what we have done is we took a 10-mer series like this with the 3 80 regions, 80 regions in the middle, and this contains 4 80 sequences, and you can have even GC sequences we tried. So basically to find out whether it binds with the DNA in the minor groove or in the major groove, or then find out what are the specific interactions, what will be the energy of the energetics of the interaction. All these things we want to explore from these studies. So first we did dock it with some DNA molecules and the small stretch of DNA and the curcumin. And when they docked in the minor groove, we used flexi docking, so it could go anywhere, but the compound the molecule selected to bind in the minor groove. So we have a lot of confirmations, and we tried many possibilities to get the best structure, okay, best dynamics. So what we have done is 80 GC and 6 80 sequences in the middle of the DNA, 4 80 sequences in the middle of the DNA, and for GC sequences we took and we did the simulation. One shows the DNA alone, other one shows the DNA, the complex, and other one shows the drug alone, that is curcumin alone. So in all these cases, you find that it's reasonably good from the point of view of the molecular dynamics. So the thing is that even for the entire stretch of the molecular dynamics, the drug binds in the minor groove of the DNA sequence very firmly. That is quite important. Okay, this shows a small stretch of the interaction between them, the small stretch of DNA trajectory. You find that even if you shake the molecule violently, still the curcumin molecule binds in the minor groove very firmly. So this is a small stretch of DNA, a small stretch of the trajectory. So you can have reasonably good computational methods which is available to find out, extract the details from that. So when you analyze it, what you find is weak hydrogen bonds are there formed between the ligand and the DNA atoms in the minor groove. And you find water-mediated interactions and van der Waals interaction. These are the major contributions to keep the molecule in the drug molecule in the minor groove. So if you look at, compare with the crystallographic details, crystallographic structures, you find some water molecules. You see that? The black balls are the water molecules in the minor groove. So what they do is they don't sit there just like that. They make mediated water contacts with the protein, the DNA molecule, and atoms in the DNA molecule and the drug. So basically, the drug molecules binds in the minor groove, replacing the water molecule. Normally, the DNA molecule is surrounded by a lot of water molecules to make it functional, and water is essential for that. So in the groove, when it binds, most of the water molecules are replaced. The remaining water molecules do have specific roles in keeping the curcumin molecule in the minor groove. So this is what you find some similar to crystallography, contouring the electron density. When you calculate, you find that this is the minor groove where this one sits. One is 80 rich sequence, other one is 84 sequence, and this is the GC sequence. In all these cases, binding is quite favorable. Now you can calculate the binding free energy, how much the energetics of the binding. You can have all these parameters. Finally, what it comes to us is the delta G. Gives free energy, how much change is there in binding due to the binding. So you find that in when you have 6 80 sequences in the middle of the DNA sequence, then you have the energy change is given by minus 13.71 kilocalorie per mole. And for 84 and 6, both you have 6, you have 2 conformations, but 84 you have another one. But GC also somewhat similar value, which I did not put here. But there is no specificity difference in the energy for 80 or GC sequences. Okay, that's what I would like to explain now, and then I will go to the piperine molecule extracted from pepper. We did similar kind of calculations. The molecules parameters were standardized using the Gaussian program. Even for curcumin, we did that. Finally, the same procedure what we have followed for the curcumin is followed here, and dynamics is performed. You find that two type of both these simulations will tell you, one with 86 regions, 80 sequence in the middle of the DNA, and the second one with GC sequence in the middle of the DNA. In both cases, it stays very well throughout the stretch of the simulation. So normally, if it doesn't bind, if the energetics is not favorable, this will come out. At least partially, the molecule will come out. Doesn't stay in that. Finally, they will become separated each other. They will be separated each other without having any hydrogen bonds or any van der Waals or any stacking or any type of interactions. Here, this shows that the molecule can stay very well in the molecule and is in the groove of the molecule, DNA molecule, and is energetically should be favorable. I'll go to the next slide. You find that, okay, this is a overlay of several conformations of the DNA and overlay of several conformations of the piperine molecule. In these two cases, you find that it binds over here with the 3 AT rich sequence, AT sequence here, the shaded ones. And you have very specific interactions between the drug molecule and the DNA atoms. So again, this water molecules, actually it is when it binds in the groove, as I said earlier, the water molecules are replaced, and the remaining water molecules do have specific roles in keeping the structure of the complex together. Okay, I will compare the energetics later, but if you, we have done also some other simulations for studying the expanded DNA sequences. The people are looking for the alternate genetic systems, keeping the Watson-Crick surface intact. So what we have done is we expanded the DNA base by introducing a benzene spacer, and it's very difficult to crystallize. I don't think that anybody has crystallized so far. We did it a few years back, and some people in US is also some elsewhere in the world is also trying, but none of us could crystallize. But DNA structures, NMR structures is also available. But to make a comparison of this with the NMR structure, we did this. This is a simulation which I don't want to show. Okay, this type of calculations will become very useful if some molecules which you don't want to crystallize, or they don't want to crystallize itself, or it may take a lot of time, or it's almost impossible to crystallize. You have a way to find out the exact structure of the molecule. Whatever the structure we got here is it compares with the NMR structure, but you know that NMR also has a lot of difficulties, ambiguities. Of course, maybe this method also has equally difficult, equally ambiguous, but put together, you get reasonably good estimate of the structure. Anyway, this came in as a cover story in the biopolymers in one of the volumes. So if you look at the biophysical approaches to study molecular interactions, so far what we have considered is the computational approach. Then we will look at the biophysical approach to study the molecular interactions and how do they compare each other. So we can use UV visible, fluorescence spectroscopy, melting temperature, isothermal titration calorimetry, differential scanning calorimetry, and electrophoresis. These are the techniques which we have used. Some other is also available. So this is the UV visible spectroscopy. If you look at the fluorescence of this is curcumin, DNA with curcumin. So you see that when you add the drug molecules, you have the concentration which changes. From this, we can find out the binding constant. The strength of the binding, this is calculated as 2.58, 6.39, or 8.22 at different temperatures. This is the temperature dependent UV visible absorption spectra of curcumin. It shows nearly around 420, a little above 420. That varies with the different concentrations of curcumin. So this is one of the way to look at the binding propensity. Then you have This is for curcumin. Again, curcumin shows that this inset shows the variations in the curcumin. Okay, this is what the 260 nm shows the DNA. But here also you can calculate the binding constant, which is 2.06 into 10 to the power 4. This is for two different temperatures. Okay, both these compares with the values what we got even for the curcumin. I mean, calculated from the experiment theoretical calculations, whatever we got is almost similar to this value. And of course, this is also found the cover story in under the Spectrochemica Act back volume. Okay, then this is just the temperature binding of piperin with DNA. This is the reverse titration. So you take the concentration of DNA as constant and increase the concentration of the drug till you get some kind of variation in the absorption peak. Then you do the reverse titration, then also you can calculate same kind of response is observed and you can calculate the binding constant. Then this is the fluorescence. Of course, when a drug binds, when the drug binds, there is change in fluorescence of the drug. Fortunately, the DNA doesn't have any fluorescence. So the fluorescence quenching, the fluorescence variations, if you look at the fluorescence variations of the drug molecule, you can always estimate the binding constant. So here the binding constant calculated is of the order of 10 to the power 3 per mole. Okay, then you can always use this isothermal titration calorimetry. That's one of the popular methods to calculate the energetics of drug binding. So you get a lot of parameters from which, so the technique I don't want to explain, but what the results you are going to get is the thermodynamic parameters. The most important one is the binding free energy, which is I think it blocks 6.3 something, 6.6 something kilocalorie per mole. This is what we have seen earlier for curcumin and piperin. If you look at another method, this is what is differential scanning calorimetry. So the basis of differential scanning calorimetry is that if the drug binds in the minor groove, it's very weak binding. If it binds in the intercalates between the base pairs, if any kind of drug which stacks between the base pairs, then the binding is strong. It's very difficult to melt it. To come out the drug, you have to give more energy. Otherwise, it doesn't come out. If low energy is enough to make the drug out of the groove, minor groove of the compound, or minor groove of the DNA. So the red one shows the DNA with curcumin, and the green one, the blue one shows the DNA only. There is not much change in the calorimetric absorption at the melting. So there won't be any, this doesn't find any major changes. The melting temperature found is 87.1, and the other case it is 88.1. The melting by means, what we understand is that 50% of the DNA separation takes place. The duplex DNA will split up into single strands, almost 50%. This is what the calculation says in differential scanning calorimetry. So since the temperature difference is not much, we can easily assume that this is the groove binder. Curcumin and piperin are the groove binders. So another way of doing it is the melting analysis. So that is, if you look at the absorbance of the DNA at some particular concentration of the drug, DNA and the drug at some particular concentration, you measure the absorbance with increase of temperature. So at the melting temperature, when it melts, you find that there should be a very large absorption of energy, especially something like 7 to 8 degrees Celsius difference will be there for a groove binder, not a groove binder, but for a intercalator. But for the groove binder, it will be something like 1 or 2 degrees. So in this case, you find only a very minor changes. See, if you look at the TM value with 84.45, when you add 10 micromolar of curcumin, you find 85.29. If it is increased 20%, you find only 20 micromolar. It's only 84.78. It shows that, again, it's a groove binder. Again, you can calculate the binding constant. All those things you can do, but all this comes within the limit of experimental errors. Agrees, all these different kind of experiments agree each other. You can also do electrophoresis. So the first well shows that the curcumin only, DNA and curcumin. This is DNA only. Second one is added curcumin added at various concentrations. You find the difference. So since it becomes complex, it becomes it's finding it difficult. The mass increases or the conformational overall conformation of the ctDNA is not a small stretch of DNA. It is a half-timers DNA. It's a huge molecule complex with the DNA. Then you find that the width of the column, the separation between the added one and the sample migrated through the column will tell you whether there is binding or not. So since it has only one column at the bottom, it's what type of binding? We assume that it is groove binding. Otherwise, you could have different variety of conformations. You know that if there is an intercalator, then there is a hinge, a huge amount of hinge, something like 20 to 30 degrees is involved in DNA. So therefore, a major conformational change in the overall conformation of ctDNA is expected, which is not available here. So we presume that it is again groove binding. Now, I will compare with the, of course, I don't have the many details of fluorogenic acid, which is binding with the silver and nanoparticles and gold nanoparticles. What we have done is a similar kind of drug functionalized with gold and silver nanoparticles functionalized with fluorogenic acid, which is plenty available in coffee and cereals. We consume it every day. So this is the molecule which will be adsorbed on the surface of the nanoparticles. We'll find out what kind of binding this can give you. So this is a nanoparticle silver. This is for the gold. You have the XRD pattern, says both are FCC structures, and you have spherical shapes, not exactly spherical, dodecamer shape. This is the EDS spectrum. All these corresponds to whatever the structural information is available in the literature. So then what we have done is, okay, this is a pure characterization of that. Even UV characterization will tell you the same absorbance for gold and silver. And again, the particle size, FTIR, all those things are just to characterize whether the particle formed is silver and gold. Now, complex with the gold and silver nanoparticles. So then what? You don't find very many changes in the XRD pattern, but you find some changes in the some kind of agglomeration in the okay, this is the okay, this is the one. Let me see. I don't have it there. Okay, I did not have. Okay, fine. When it's complex, we have all these analysis, but there's some kind of the agglomeration is observed because of the surface functionality. Then you find UV visible absorption. This is what you get for the DNA, complexation with the DNA. This is again, you find that addition of this drug, functionalized DNA, changes the absorbance of the DNA. This itself shows that there is binding. And again, you find that this third figure shows the absorbance of gold nanoparticles. With addition of DNA, it changes. Here it shows the silver nanoparticle there also. Whatever the absorbance of silver and gold changes with the addition of DNA. So you can calculate the binding energies. You find that 3.1 into 10 to the power 4 per mole per Kelvin per mole. Anyway, you find the kilojoules per mole. So in kilocalories per mole. In both cases, you find that the values are acceptable with the experimental observations. And here in fluorescence, quenching is also observed. The previous two were absorbance measurements, but here we can have the fluorescence measurements. You find the addition of DNA quenches the fluorescence of that. So from here also we can calculate the binding constant that will tell you the propensity of binding between the two complexes. We find that the values which we got are in the acceptable range. And again, to look at the conformational changes due to the drug nanoparticle complex in the DNA groove, what we have done is a CD spectroscopy, but actually you don't find very many changes in this around 245. That will tell you the alpha helicity, how much it changes, that or how much the DNA unwinds. Here in this case also, we don't find any major changes, less than 10% unwinding of the helicity occurs. That is quite normal, and the DNA will be functional. This gives you ITC calculations, whatever you have done for earlier. Here also you find that the binding energy, delta G, change in binding energy is minus 7.43 up to minus 8.52. All these will tell you that you can monitor, you can find out the exact binding nature of the molecules, whether it is bigger ones or smaller ones in the groove, the DNA or not. So I think my chairman is staring at me, so I would like to stop here. So there is a possibility that you can compute. You can have calculations. If you cannot crystallize or do any other kind of work, you can calculate by molecular dynamics. You can do spectroscopic analysis, various spectroscopic and calorimetric analysis. One thing that you make sure that the experimental results that you get are consistent or not with various different type of experimental techniques. The experimental techniques that we have used are a variety of techniques, but all of them will lead you to the same kind of results. That is quite interesting, and this is what you expect to have. Okay, finally coming to the acknowledgements, I thank my students and also the Fatima College, my own college where I studied, and my the people over there for giving me an opportunity to come over here, and the chairman for your kind words of introduction. Thank you for your patient listening. If you have any questions, I would like to answer.
Thank you very much, sir, for your valuable presentations. It was very interesting. Words of a structural biologist, I would say. I'm sure you can have questions now, two or three questions. So I request the audience to come forward and share your thoughts with Professor Sadashan Kumar, sir. Please.
Any of the students who wants to ask questions, even later, put my in my email, you can ask me.
Sure.
Any questions from the audience or especially the students, you can ask me through email also, even later stage if you want to ask.
Yes. Any questions from the audience, please? Please come forward and share your thoughts.
Again, if there are no students asking questions, I have one question to you, Professor Sudarshan. This is Saji George.
Please,
if you allow me.
Sir. Yeah, please.
Okay. First of all, we cannot see you. I think your camera is covered. We would have appreciated.
Really?
See us. And second thing,
actually, okay, is there? Can you see me?
It seems like your camera has some cover over, so maybe if you remove the cover, you probably can see. Anyway, that's not the major thing. My question was like, curcumin, when it goes through the digestive tract, it may change in structure, like especially hydration can happen. So do you think that kind of structural changes will affect the binding? I presume it may, but what will be, how do you think that hydration of the molecule, because it's a hydrophobic molecule, extremely hydrophobic molecule.
Sure. It is very much possible. We don't know. One thing I can tell you, DNA is very stable at the presence of water. At any amount of water, it can hold. Basically, it holds on the grooves and the phosphate backbone. So I think it will be filled with maximum capacity, even if it goes through the alimentary tract canal, or food pipe. But maybe, I don't know, but anyway, people use it for drug delivery system. So I don't think there is much changes that could happen.
Okay.
It is very, very much possible, which what you suggested is possible. But I don't think if it does that, our conclusions will be different.
Yeah, I just to point out that. So it seems like actually hydrate.
But actually, our molecular dynamic system will also put in hydration only. We set up a thermal water box tip 3P. We call it as typically called tip 3P water molecules. So if the DNA is there, the entire system is there put in a box of 11 angstrom over and above the surface. Then we do simulation. So water molecules can move. We'll put some thermostat also, put it inside a number. the number of molecules, if it goes out, sometimes it goes out, equal amount will be put it inside and then do the simulation. So water, actually the water hydration has been taken care of while doing the simulation.
Great. I just want to tell you that, I mean, but it's a great work, like it's the fundamental studies looking at these kind of through the simulations and also through the biophysical methods looking at these kind of molecular interactions. And I believe that by changing the structure, we can easily fit into that simulation that you have already created. So it won't be much difficult to predict those kind of binding activity, the energy, if the molecule changes even with a couple of molecules of a water molecule. Great, thank you. Appreciate your great presentation. And
yes, thank you
so much.
Saji.
Thank you so much.
Thank you, sir. Thank you, sir. You were wonderful for your wonderful talk, for sparing time with us. Thank you very much, sir, from outside.
Great. Thank you.
Sir.
Thank you, and thank the organizers. Yeah,
with your permission, we are just moving to the last part of this fifth session. We have the plenary talk. We have Professor Saji George with us. Professor Saji George, I'm really happy to see you. Good afternoon. I'm really happy to introduce you. I'm really happy to chair this session where you are speaking because you are our own student. Proud of you, Saji. Yes. Hello, Saji is there? Hello.
Yes, yes, I'm here. Yes, yeah.
Yes, I'm really happy to see you. I'm really happy to introduce you. I'm really happy to chair a session where you are speaking. Right. Very nice.
Great.
Because Saji is our own student, a student of School of Biosciences, UMG University. Saji, good afternoon.
Thank you.
Yes, very good. Nice to see you in this platform of this international conference. Yeah, from outside, from the whole School of Biosciences side, we congratulate you for all your achievements, for reminding us a very, very good, ideal model for our students and for everything. Anyway, we are here for the talk naming the Nano Giant for Food Safety and Security. We have Professor Saji George with us, the Professor of Department of Food Science and Agriculture University, McGill University, Canada. He took his BSc in Food Science and Quality Control, and he took his MSc in Biotechnology from our own department, School of Biosciences, Mahatma Gandhi University. And after that, he took his PhD from NUS, prestigious National University of Singapore, one of the top 10 universities in the world. And then he took his postdoctoral training in nanotoxicology from University of California, USA. And then he joined at McGill University in 2017. And Dr. George was heading the Center for Sustainable Nanotechnology at Nanyang Polytechnic, Singapore, from where he completed several research projects addressing the health risk of nanomaterials and environmental contaminants. He has more than 70 publications, 37 H-Index, 7 patents, many awards, and he is an excellent teacher, I know. So with all this, I welcome Professor Saji to deliver the plenary talk. Dr. Saji, please.
Thank you, Professor Jayachandran. It's always a pleasure to be introduced by a person who is a real inspiration for many, including me. He has been motivation for me to continue my journey in academics and research. And before I start with my presentation, I would also thank organizers, especially the chair, Dr. Jean George, for inviting me to give this plenary talk. As well as other members like Dr. Sunil Thomas, who've been constantly contacting me to make sure everything runs smoothly. And all the listeners, welcome to this session where I will be happy to share some of my research on nanomaterials. And like Dr. Jayachandran introduced me, my study has been mostly on the nano-bio interactions. So, but I think that has a high relevance in securing food safety and security, food safe security. Let me introduce to you, before I start with my lecture talk, let me introduce to you the evolution of tools that are used in agriculture, right from the use of animals through the use of machineries And now we have reached a stage where it is the convergence of many of the technologies. It's not just one or two tools now. It's a convergence of many technologies coming together to realize wonderful things. Like, for example, now we have the tools in terms of nanotechnology that started early 2000s or before, but mostly in the case of food and agriculture, later part of 2000. And now we have metabolic engineering. That's another biotechnology advancement that has materializing many of those unthinkable things earlier. Now, with primary products were the harvest of earlier kind of technologies and tools. Now we have come to a stage where we can create meat in the lab or in a fermenter. And also we have products recently released news on the dairy products like milk or cheese that is entirely produced from transformed bacteria, for example, as well as we already know that the commercial success of Impossible meat, which is basically a simulated meat or a meat analog that is entirely produced from plants. And these are happening at this right juncture, and there is a major transition that is happening in food because of the use of new tools apart from the beast or the animals that we used to plow the field. Instead, now we are actually using these kind of advanced tools. But the spirit remains the same. We need to make sure that the tools or the techniques that we are using has been tamed well to give the benefit instead of going out of the way or out of the intended purpose and giving us some harmful or unexpected adverse effects. So that is the bigger picture of my talk. So with that nice tone, let me introduce my group. I'm heading a group, I call it as a Sustainable Agriculture, Food and Environmental Applications of Nano. And when you abbreviate it, it is Safe Nano. So, and also we are promoting that safety and sustainability of nanotechnology. And we focus three aspects. One is to understand how the nanomaterial interaction with biological system will lead to some toxic effect. That is, if you were to tame or to use that tool for a useful purpose, especially in agriculture, we want to make sure the technology that we are developing is safe. And we need to develop those kind of platforms that can tell us about the safety aspect. Now, we heard from a professor from Leeds talking about the toxicity of materials, especially for the female reproductive system. Similarly, we use many of those newer platforms, mainly based on adverse outcome pathway, in order to check whether the physicochemical property of nanomaterial has any influence, or what is this relationship between potential adverse outcome in human, that we use high content screening or high throughput analysis. That's the newer platforms. And we use that knowledge gathered from there in order to develop sustainable nanotechnology for various purposes, including antimicrobial strategies, or infection control using vaccine delivery, or infection control using what we call as nano-enabled antimicrobial combination therapy. And many of these techniques or many of these developments from our lab, I will be happy to share with you. And also we work with industry, some translational technology for developing nanomaterials that can be useful for example, nutraceutical industry or for food industry. I will be happy. I will be sharing some of those developments from my lab today. So, for those of you who may not know much about nanotechnology, let me briefly introduce what is nanotechnology. By definition, it's the manipulation of matter at the nanoscale to generate size-dependent functionalities that are different from atoms or bulk. Keywords are size-dependent functionalities that are different. So nanomaterials are materials with at least one of their dimensions between 1 to 100 nanometer. That is a very rudimentary or elementary kind of definition of nanomaterial. It's not a functional definition, but that is useful for getting an idea of what's happening. The point is, like, when the materials are in nano, for example, they behave differently from their bulk counterpart. For example, we know carbon that manifests as a dull carbon dark material, which is an insulator, which means they are non-conducting. Or carbon, when the atoms are arranged in specific order, we get diamond. Or we get, for example, when it comes to the nanotechnology, we can actually arrange these atoms, or in a specific way they are arranged, they will give rise to, for example, if there's 60 atoms of carbon arranged in a spherical form, we call a buckyball, or a carbon C60, or if it's forming a single sheet of that carbon covalent bond, and we call it as a graphene. And when you roll that graphene sheet, you get carbon nano 2, although the process is not entirely like that, but we can imagine like that. Now, all I want to emphasize is when these materials goes in the nano dimension, they achieve some of the extraordinary properties in terms of their behavior. For example, diamond, that we know as a brittle, hardest natural material, it cannot be, you cannot bend it. It will, once you try to bend it by excessive force, it will only break. But as we know that if you grow the diamonds of the nano forms of diamond, you can apply force that will behave like a elastic material, which is because of extremely smooth surface. And the carbon nano 2s, for example, which is made from carbon, has extraordinary strength as well as conductive properties. And we also know that metals like gold or silver, you can make it into plasmonic, which means that because of their unique electronic configuration, their interaction with electromagnetic rays can give rise to size-dependent color formation as we perceive outside. And there is a lot of things that is going inside why they behave, because where these nanomaterials are sitting in the transition, in the boundary between the Newtonian physics that applies to the bulk material, as well as the quantum physics that applies to molecules or atoms. So being in that intermediary state, they do have a dominance of quantum effect and partially behave the Newtonian physics, for example. So, and along with that, they have extremely high surface area, extremely high number of atoms or proportion of atoms or molecules that are exposed to the outside in comparison to a bulk material, etc. That gives them unique property that has been utilized for many applications in the electronics industry, in many other sectors, including chemical industry, photoactive materials, etc., etc. Now, that unique property can also be put in function when they interact with the biological system. As represented here, there is size and shape dependent biological responses that are generated, because as we know that biological system was not engineered to face engineered nanomaterial, or biological system has not evolved to accommodate engineered nanomaterial, although many of those biological functionalities are taking place at a nanoscale. And as a result, we will see unprecedented kind of responses when we put nanomaterial. For example, just now, Dr. Sudarshanan presented about the minor and the major groups of DNA. And you see, when the gold nanoparticle is made it into a size below 5 nanometer or so, they can actually go and intercalate between the major group, and it can give rise to DNA breakage, and then that forms the basis of DNA gold nanomaterials in cancer therapy. And so there are those kind of unique interactions that can happen. There is surface chemistry, core chemistry, and then there is multifunctionalities. All these are some of those aspects we will come across. Now, these improved or novel properties presented from nano has, like I mentioned, diverse application in different sectors. And there is a lot of application in food and agriculture sector, which you might not have imagined. It starts right from the production of food, from the food security point. That means you can improve the productivity of crop by using nanofertilizers, on-demand pesticides that becomes only activated on contact with a pest, so that safety is ensured. And then vaccine delivery in aquaculture, in animal agriculture, antibacterial technologies in animal agriculture, as we will see later on. And through the nanomaterials that are used in the food processing side, for example, high hydrophobic surfaces that repels dirt and water, so they remains clean always, away from, clean or sterile from microorganisms, to some of those flavor containing compounds or nanomaterials that are used to enhance the organoleptic perception about food. For example, the colorants, I will show you some of the examples later on. And then their use in tracking and sensing in order to see how the food has been handled, for example, by using sensors, etc., etc., and to active and intelligent packaging materials, which will tell you whether the food is wholesome and good or safe to consume without having to look at the expiry date, because it is directly interacting with the environment in which the food is packaged. For example, a fruit or any other food is packaged. So it is sensing from the gases that is released, and so it tells us whether the food is good to go or not. And so much so that we will think these are kind of hypothetical kind of situations. No, it is not. That kind of technologies are available in the market as and when we are talking about, and it's almost $1 trillion business. Obviously, when there is $1 trillion business, these kind of materials will show up in our plate and we will be exposed to that. So when if I ask you, do you have consumed anything, you may not say you not have consumed nanomaterials, but in fact you are. So now that we are dealing with a technology that we are going to put into our body, because I will show you some of the examples, we need to make sure that they are safe and sustainable. So if you look for sustainability, or sustainability, that terminology has a major three aspects. That is, first one, it should be safe to human and environment. It should be economically feasible. That means a technology that is developed which cannot be translated for industry, it will not be viable. And it should also be socially acceptable. People have to accept it, embrace it. Otherwise, that technology will not be viable. So if you look for an icon for that kind of a technology, you can come across this pencil here. It's a simple device, simple, economically feasible, it's a safe. And more important thing that I want to highlight is the presence of this eraser on this pencil. And why is that eraser there? It's because it's an inbuilt design to make sure that if there is any mistake that happens from that pencil, there is an inbuilt design to erase it. So metaphorically, we can look into that as a safe by design strategy, or an inbuilt safety feature is there. So if we had that kind of idea keeping in our mind when we develop a technology, we may not have that kind of issues, something like what we are facing now, like the plastic industry with the microplastics and nanoplastic polluting all the environment, for example. So there are some, with that thought, let me introduce to you, this forms the basic principle of many of our technologies that we develop in our lab. So with that, let me introduce to you some of those aspects. So here, the first aspect that I want to give you is about the potential toxicity of nanomaterials that are gone into food. So here is an example of titanium dioxide and silicon dioxide and silver. Titanium dioxide, as you may be thinking, why are we talking about titanium dioxide in the context of food? I have to understand that many of those food that we consume, titanium dioxide are added as a white pigment. There is no good alternative to titanium dioxide. The same material that is used in the walls as a paint is used in our food to give the good masking power. And obviously, it is not all of them are not nanoparticles, but a substantial portion, about 37% of food grade titanium dioxide, is in the definition of nano. Similarly, there is another material that is used, is the silicon dioxide, that is used for many of those manufactured products, because it improves the flowability of the food, because it absorbs the moisture and keep the food particles separated so that it can be nicely flowing for dispersion processes. And as you may know, some of those, you may be familiar with this artificial sweetener package, 99% of which is silicon dioxide in many cases, not always. But as you know, the active ingredient is only forming around 1% or less than 1%. So, and another technology that is coming up is the silver nanomaterials that are used in food packaging, and as you know, is also may also get into our food. So we need to look into what are the potential toxicity. And we know that these materials have been used, especially titanium and silicon dioxide have been used from the middle of last century. So it's been used for a while. So we know that nobody is dying immediately of consuming these food products containing these. So we know there is no acute toxicity. In toxicology, we say acute, which means we don't see an immediate effect. But does not mean that they are biologically inert. So we have done some studies looking into those kind of materials that are we consuming. And first study that I want to highlight is what happens when this material goes into the food. Here is an example what shows that we use some materials like titanium and silicon nanoparticles that are interacted with milk, an example of a food matrix, and we retrieve those particles, look them on the surfaces, what is there on the surfaces, using some of the advanced imaging techniques, X-ray fluorescence based techniques, and we could see that their surface, for example, here is a titanium dioxide given in green, and then there is we can take the signals of lipids as well as protein and as well as carbohydrate in this material. So which means once the material goes in, they are immediately covered with these kind of biomolecules that are present in the food. We can call that as a corona. It's called as a surface corona. Now, what we did is we looked into the surface corona for the protein concent, the type and amount of type proteins that are getting absorbed onto the surface. Interestingly, what we find is some of those, we did a lot of study to understand what is the kind of bound onto that primary molecule or the primary layer, which probably is the lipid layer. And then there is a lot of protein-protein interactions we could see, protein-lipid interactions, etc., that are playing a major role in shaping that. Now, interesting part is some of those proteins that are relevant for food allergy, especially, milk is a cause of milk allergy. Around 2% of population in this part of the world is allergic to milk. And we can see there is a magnification and an enrichment of those kind of proteins on the surface. And that is something really concerning. Why? I will tell you with our studies and other studies that I will exemplify. Why is this a concern? Like, when nanomaterials are entering our body, they are not concentrated, or they will not be confined to the primary target organ in which they are exposed to. For example, if you are taking through the food, they are going to our gut and then the intestine. They will not be confined to the intestine. Through the lymphatic as well as circulatory system, they can gain entry into the body. And since they are small, like I mentioned, they can creep into, or they can creep across many of those biological barriers to reach distant organs. For example, there have been studies shown that inhaled particle as well as ingested, that is eaten particles, can be found even in brain, liver, kidney, etc., etc. And like I said, now when they are traveling, because they have gathered a surface of molecules, they're not traveling alone. They're actually taking that cargo with them. They're traveling, they're taking those kind of allergens, those kind of toxicants they are present in the environmental matrix. They are traveling with that cargo, and they are delivering that cargo to the cells. And so as a result, there could be potential toxicity. So if you want to study those kind of toxicity, we thought it's a good idea to look from the molecular interaction, because when a toxicant enters the body, the first step is they start with a molecule. They interact with a molecule, alter the shape and size of the molecule, especially protein and DNA. So we saw from Dr. Sudarshan's presentation how the molecules can alter the structure of a DNA, for example. That's a molecular interaction. There are toxicants that can alter those kind of structure of protein or DNA. And so that is a molecular initiating event. Or it may be inducing, for example, radicals, free radicals, for example. Those are called molecular initiating events. Now that molecular initiating event then progress on to cellular responses, stress responses. It can be like an oxidative stress. It can be like an inflammation, for example, that the cells actually receive that stress and they respond through activating certain genes or through other mechanisms. And that don't stop there. They may propagate to the next level at an organ level or a tissue level, where there is a failure of the tissue to function properly, and then finally that will be seen as a disease or a pathological condition at an organism level. And when it continues for a while, we will see there is a wiping out of population of, as we have seen with the DDT, for example, wiping out the populations of birds, for example, in California, or silver that was used in film industry from Hollywood, wiping out the population of some of the clams, river clams in California, etc., etc. Some of the best examples how that molecular initiating event propagated through different stages or different hierarchy in biology to reach or to have an effect in the population. So with that, what are the kind of molecular initiating events that we see with the nanoparticles that are going in food? So here is an example of nanoparticle like titanium dioxide or silica particle here interacting with the digestive enzymes. For example, here it is a trypsin, and we can see the structural change in trypsin using CD, that is circular dichorism. So we can see how much alteration in its in the secondary structure is happening because of that interaction. And we can also calculate or also assess the inhibition of enzyme activity, and we find that there is a partial loss of trypsin activity because of its interaction with nanoparticles, but not with microparticles, and that will lead to partial digestion of the substrate. For example, in this case, albumin used as a substrate, which means that when you are consuming food with nanoparticles, it may interfere with some of the digestive enzymes so as to have a nutritional function of our food compromised. And that can be seen with other enzymes like amylase, salivary amylase. We have shown that and published this result, again showing that there is an alteration in the structure of the protein, which can be seen as a adverse effect. Then I also told you about the interaction of proteins, because when they goes into the milk, they actually accumulate some of those proteins. So what we were interested to see if they will change the structure of those proteins that have allergic responses. And what we find is, yes, again, this is a FTIR spectrum showing how the secondary structure of the protein here is a lactoglobulin, which is a protein implied in food allergy, and we can see that change in structure again as a free thiol group, because there is interactions that alters the structure of the protein, and that has a effect on master cells. So we tested against human master cells that will respond to this allergen by priming the master cell before with a human sera, and we know that these interaction, this change in the structure of the protein enhances the allergenicity of that protein, meaning to say that that molecular interaction that is happening alters the structure of the protein, and that alteration is in fact increasing the allergic response. So that is not good. So from that change in the protein structure itself, we know that they are changing the allergenicity. Now I told you, like, once they are taken, or once these kind of nanofilm or being surface corona is formed, they will facilitate the uptake of that protein across the epithelial layer. So we wanted to see if that happens. So once we consume the food, we know it goes into the intestine, and intestine has a safely guarded kind of cells. There is an epithelial layer which is tightly bound together, and there are other cells of relevance to immunology that is guarding our the defense in the intestine, making sure that no obnoxious or foreign matter get into the body unless it is intended to. And so, there should not, these are called tight junctions. So we were interested to see how the nanoparticle, when they are interacting with epithelial tight junctions and adherent junctions, if they have any effect. And in fact, we found that nanoparticles, again, not with micron particles, we can see these tightly controlled or tightly bound or adhering cells now get separated where in the presence of nanoparticles, you can see it as in confocal microscopy as this gap between the cells are opening up, as in the case of titanium dioxide, or with a positive control here, or with even silver nanoparticle, we can see the same kind of effect. We can also we can quantify that using by measuring the electrical resistance across that epithelial layer, and you can see as a function of concentrations of these nanoparticles, especially with the silver, or with a higher concentration of silica, or with a higher concentration of titanium dioxide, there is that drop in the resistance, which is also an indication that there is an opening up of the gap between the cells. And we can look in why that caused. We can look into the molecular interactions between the proteins, and we can see that some of those proteins that are involved in that tight junction as well as in the adherent junctions are affected. Especially what we see is with the cationically charged silver particles, all these classes of proteins are affected, which means they are retrieving into the cytosol, which means they are no more adhering properly. But with the silica and titanium dioxide, we see a major effect on the tight junction, but much less effect on the adherent junction, which is the ecadheric. But they are not benign to any of those. And so we can see when you look into their, possibly we can see there is actually happening as a function of oxidative stress. That's another thing that we wanted to see. And as a result of oxidative stress, there is an inflammation. There is inflammatory markers, and we believe that in the case of silica and titanium dioxide, it is that inflammatory markers that actually causes that retrieval of those protein that makes that junction tight. Now, in the case of silver, there are additional mechanisms which actually make sure that it can open up. When they open up, like I mentioned, if that junctions are not tightly guarded, there is possibility of seepage of unwanted things. And we saw that allergens, food allergens are not supposed to pass through. When you consume milk, for example, milk is supposed to be digested properly. And if it is digested, and again, it is the amino, it is absorbed in the body as amino acids or as smaller peptides, not as the lactoglobulin as an intact molecule. Now, when these tight junctions are compromised, we will see that as we did some of the experiment with a co-culture model here, transwell assay, where we have an epithelial layer on the top and then mast cells on the bottom, and we challenge that epithelial layer with the nanoparticles, and we see the availability of lactoglobulin, for example, has increased in comparison to negative control. There is a substantial, significant increase in the bioavailability of those proteins in the mass, in the basal compartment, which means they percolated or passed through those epithelial layer. And then they also causes this increase in the mast cell degranulation, meaning to say that a surface coating of those proteins actually decreases their digestibility and also increases their bioavailability across the epithelial layer so as to enhance an allergic response. So these studies are published now, and it's the first kind of study showing that these kind of possibilities. And we argue that some of those increase in the allergy, food allergy that we see with the populations who are switching to Western food is partially because of this reason, and more studies are going on. So is there, like I said, we are talking about sustainable nanotechnology applications as well. So we also looked into the possibility of creating something alternative to titanium dioxide. And here is an example showing that we created a composite material containing layered double hydroxide, which is a synthetic clay, for example, where we intercalated it with the proteins and then polymers of carbohydrate, and you can see they have a good masking power. They are not as comparable as the titanium dioxide, but they are superior in its performance in comparison to many of the other commercially available alternative. And we show that it has a good masking power. As you can see, here is the titanium dioxide. Here are the composite that we generated, and which we think has a greater potential because we look at the safety, efficiency, and cost, and we decide that our particles that we generated has a very good sustainability index, what we call here. So that is the key word here. It's not just looking at the safety aspect or looking at the performance in terms of efficiency, but we also look into the cost so that it can be translated. And now there is an innovation disclosure file, and this paper is published now. And another, the other part I want to emphasize is on the silver nanotechnology. Like I mentioned, silver is used extensively in food applications, especially in food packaging, for making sure that the material that is stored stays longer, because silver has an antimicrobial action. So, and also silver is used extensively in animal and poultry agriculture, because as an alternative to antibiotic, in order to take care of the microbial infections. Now, what we see is the silver has the bacteria like multi-drug resistant staphylococcus or methicillin resistant staphylococcus, for example. They also resist silver because of the same functions of efflux pump. And we found that there is an activation of those kind of efflux pump when you prime the bacteria cells with a smaller concentration of silver. And we also looked into molecules that can inhibit, and we found that there are molecules that can inhibit the function of efflux. And based on that knowledge, we regenerated or redesigned silver nanoparticle, which can either go and bind with the bacteria. What we see is the bacteria cell nicely decorated with our silver nanoparticle in comparison to a silver nanoparticle which is negatively charged, and so it enhances the bacteria killing. And another possibility is co-delivery of these efflux pump inhibitors with the silver, which also improve the antibacterial property. Now, from that, inspired by that, we actually created a new kind of a therapeutic, which is nano-enabled antimicrobial combination therapy, by combining more than one drug using nano. Like I showed you in the beginning, multifunctionality is one of the advantages that is provided by nanotechnology. So we can use more than one drug and deliver at the same time to a target like a bacteria. And so here is an exercise where we did with a multi-drug resistant salmonella, which is causing problems in animal farms. And we find the molecules or drug molecules that can synergistically interact with the bacteria. And then we put them into nanomaterials, and we show that this nano combination works better than individual molecules, or even the combination of molecules that is directly added to the suspension. And that's because when they are in the nano form, they reaches the bacteria surface together and deliver these two molecules at the same time to create a good antibacterial strategy, even targeting the intracellular pathogens like Salmonella, for example. And it can be the same technology, can be same concept. Here it is a clay particle where we in situ generated the silver particle on top with the tannic acid in the interior portion. And then we can see there's a combined effect of this tannic acid, the free molecules of tannic acid and silver, which creates a better antibacterial property, again, against a multi-drug resistant bacteria, which can be put into packaging molecule, for example, packaging matrices, for example, to give a good antibacterial responses. Again, this is also an innovation disclosure. I'm not going in detail with the another study which we've been doing, again, with the vaccination of fish for aquaculture. Again, the only part I want to highlight is when we look into that, we are not just looking at the efficiency, but also looking at the safety of those generator particles, and then the cost of it, so that we can actually identify the best suited solution for a agriculture purpose. So what I've been telling you for, introducing you here is the concept of the possibility of using nanotechnology, which has tremendous applications in food safety and security, like I showed you, to ward off against infections, to ward off against unwanted microorganisms in our food, and also to improve the productivity in terms of animal agriculture as well as plant agriculture. I have been going into plants. Some exciting studies we have done showing that the nanoparticles with the biostimulants obtained from bacteria can improve the performance in terms of the productivity of the plants as well as their growth performances. What is taming the nano giant is, by understanding their physicochemical properties, we will be able to put restrictions on a type of property that is that may give rise to adverse outcome. with that kind of design, with that kind of knowledge, we can design safety assured nanotechnology. And that is the key message that I want to give here. And I will be happy to take any questions. And before I end I would like to thank all my all my students who've been the engineers behind these wonderful data and studies, and the funding agencies and the organizers of this conference once again. Thank you.
Yes, Dr. Saji, wonderful, very impressive talk as usual. Very nice to see you presenting all these research outputs. I'm really interested in nano-enabled combination therapy. Yeah, it's wonderful, really wonderful. Thank you, Saji, once again. And dear audience, there can be a couple of questions right now. We have several appreciations coming in the chat box. Yeah. You can ask some questions. You can share your thoughts with Dr. Saji, please. Students, please come forward and you can interact with Dr. Saji now.
Hello.
Hello, sir.
Yes, please.
My name is Anjusha. I'm a research scholar at Nanotechnology University. Sir, I want to know a little bit more about this nano vaccine actually.
Yep, great. So this nano vaccine we've developed in Singapore, we had a problem with the aquaculture there. So there was the fish that we were growing, the industry was growing is barramundi. It's also called a sea bass, if you know. It's a very nice food fish. But the problem that they were facing was the infestation or infection with the bacteria that was devastating, because once that bacteria is called a Tinacebaculum maritimum, TIMA in short. That bacteria, once it strikes, it will wipe out the entire population. Almost 95% of those the fish that is grown in a facility will be gone. And the challenge was, first of all, they didn't know what is happening. So my colleagues in where I work, they isolated the bacteria, and they identified a way of growing them in the laboratory conditions. And then they inactivated the bacteria and injected it back into the fish, and you see fish can survive. The only problem, like I showed you in that slide, you have to inject it. It's like the COVID vaccination we got. Now individually we have to go and get a vaccination. Now we imagine about a million fish growing in a fish farm. You need to knock down the fish first, and individually it has to be injected with a skilled persons. And it's a painful and expensive process. So we thought, instead of the needles, why don't we create nano needles where that antigens of the bacteria that causes the disease can be placed, so it gets self-injected into the gills. And so what we did is we selected a few of those nanomaterials. We screened a library of nanomaterials, especially with the natural chemistry, in order to use it as a nano vaccine. And what we isolated the outer membrane protein of the bacteria. We put that one into these nano delivery platforms, and then we put it in the immersion vaccination, like just put these nano powders in the water where we can actually expose the fish fingerlings, which is smaller fish. And we saw that they responded very well to that, because these particles that we used had also a augmenting or a effect, as well as they had they were good in introducing the antigen into the circulatory system of the fish. And we tested for the antibody titer. It was better than the injection vaccination for some reason. And another advantage is, now we can actually immunize even smaller fish, which otherwise, if it is a injection vaccination, you need to wait till the fish reaches a certain size when you can hold the fish in your hand. So that one was a Singapore patent. Yeah. I hope I explained to you. If you have any more questions, I will be happy to introduce you to them.
Sir, then is this a kind of immunotherapy? Like how it will affect the immune system of that fish or that aqua region?
It is. It is a very interesting thing, immunotherapy. Like I introduced to you, how this nanomaterial actually alters the allergic response to food allergens. We were also looking into that possibility of developing or tuning the reaction to more advantageous, like a therapeutic ones. So, for example, if you want to develop an immunotherapy against food allergy, it may be possible by targeting specific type of cells.
It was very interesting session, and thank you so much for your response, sir. Thank you very much.
Okay. I'm sorry, I'm unable to switch on my video. Something happened in between, some technological crisis, I think. But anyway, Saji, it was very interesting. Thank you for sharing your time with us. Thank you very much. I would like to express my sincere thanks to the organizers of Fatima Mada National College, Autonomous College. And particularly, I'm thankful because this was an occasion where I could meet our student Saji also. So thanks to the audience for your patience. Thanks to the audience for your active interaction. And that's the end of session 5. Thank you very much.
Thank you.
Yes.
Great day, everyone.
Sir, thank you all. Our sessions for today have come to an end. My sincere thanks to our Honorable Chair, Professor Jayachandran K, for conducting the session. A heartfelt gratitude to all our speakers, Professor Harikrishna Verma, PR, Professor Virginia Pensabene, Dr. C. Sudarshana Kumar, and Dr. Saji George for their remarkably comprehensive and fascinating talks. This has been a very interesting and informative session. Again, I extend my gratitude to the active viewers and listeners for making this a lively event. We appreciate the efforts of all who have been a part of this event today. Before we end the meeting, I would like to give a few details about tomorrow's session. Tomorrow, we move on to the 6th session of ICMPMA 2022. The topic for tomorrow's conference is magnetic materials. The talk will start at 9:30 a.m. and end at 12:10 p.m., followed by the poster session on magnetic materials. The oral presentations will be conducted from 2 p.m. to 4 p.m., followed by the poster presentations on the topic other materials from 4 p.m. to 6 p.m. Looking forward to more knowledge and an extraordinary time tomorrow. Before leaving, we request everyone to fill the feedback form available on the chat box. Hope everyone has had a great time. Good evening and thank you to all. Have a nice day.
Okay. Thank you. Shall I leave? Thank you.
Yes, sir. Yes, sir. Thank you, sir.